for your introduction. Ladies and gentlemen, my name is Lucie Bacakova. I am from Institute of Physiology, thank you, of Czech Academy of Sciences in Prague, and our lab is focused on testing cell biomaterial interaction and tissue engineering, mainly of blood vessels, bone, and also skin. And in this case, we concentrated on potential use of adipose tissue derived stem cells, which were isolated in collaboration with the Department of Plastic Surgery, Bulovka Hospital in Prague, and we also need collaboration with various chemical and physical institutions, material engineers, private companies to prepare various scaffolds for tissue engineering. In this case, we used metallic scaffolds for bone tissue engineering and bone replacement. As for definition of stem cells, it's generally known stem cells are undifferentiated cells which can be divided to produce more stem cells, or which can differentiate into practically all cell types, bone cells, vascular cells, skin cells, neurons, and so on. And you can see potential differentiation of embryonic stem cells. We can consider embryonic stem cells as the only true stem cells really totipotent. But use of embryonic stem cells is ethically limited in human medicine, and we should search for additional sources, for example, extrafetal tissues, placenta, amniotic fluid, umbilical cord, and we can also use induced pluripotent stem cells, but they are genetically manipulated using chemical viruses, and they can be considered as dangerous, associated with the risk of immunogenicity and even cancerogenicity. And so, as the most suitable source can be considered adult stem cells. They are present in bone marrow, blood, skeletal muscle, skin, practically in all organs in the human organism, and we concentrated on adipose tissue derived stem cells. The general question is why to use stem cells, because in previous years we concentrated on differentiated cells, osteoblasts, keratinocytes, smooth muscle cells, endothelial cells for our tissue engineering. But stem cells can be advantageous because they can be isolated in relatively larger quantities than differentiated cells from patient organism. For example, they proliferate more quickly, undergo senescence later, and they are less immunogenic than differentiated cells, so they can be applied as allogenic or even xenogenic, and in the literature it is written they can act even immunosuppressively and can be used for treatment of autoimmune diseases. And important feature is stem cells are able, or larger autocrine production of growth factors and various mediators, more than differentiated cells. And as I told, adipose tissue cells are especially advantageous because adipose tissue, unfortunately, and civilized society is abundant in most patients including me, and adipose tissue is localized subcutaneous in under skin, and so stem cells are relatively easily accessible, easier than from bone marrow. And in comparison with the bone marrow, it was reported adipose tissue is richer with stem cells than bone marrow, much richer, but I read in literature, it's not my experience. And in comparison with bone marrow stem cells, adipose cells are considered to have higher proliferation capacity, later senescence, and for example, bone marrow cells are prone also to calcification, which is not very suitable for soft tissue engineering, for example, blood vessels, heart valves. In collaboration with hospital, we isolated adipose tissue stem cells by liposuction, and lipoaspirate was delivered in our lab, and we isolated stem cells by repeat rinsing with PBS in order to remove blood, because the lipoaspirate was contaminated with blood. Then we digested the tissue with collagenase, and you can see here stem cells after repeat centrifugation, and finally we filtered the cells through a cell strainer, and we seeded the cells in the quantity of 0.16 milliliters of original lipoaspirate per square centimeter. You can see stem cells, and now it is necessary to characterize stem cells by flow cytometry. Specific markers are available in literature. These markers include, for example, clusters of differentiation. They are usually part of receptors, enzymes, or these markers are related to integrin-mediated cell adhesion, or you can reveal immunoglobulin adhesion molecules. These two are markers, for example, for endothelial cells, because we isolated the cells mainly from perivascular fraction, but differentiation into endothelial cells is considered as relatively difficult. We tried the most simple differentiation to fat tissue, fat cells in specific medium containing dexamethasone, indomethacin, insulin, hydrocortisone, and you can see lipid droplets in the differentiated cells. They were stained by oil red. And we also tried osteogenic differentiation, which is considered to be relatively easy. We used special media with dexamethasone, glycerol phosphate, glutamine, ascorbic acid, vitamin D, and we stained with alizarin red calcium phosphate deposits on the cell culture substrate. Very rich staining. So they are able of osteogenic cell differentiation. But we also investigated the influence of liposuction parameters, which can influence the quantity, quality, adhesion, growth, differentiation of stem cells. For example, it is important if the liposuction is manual or vacuum machine. It is important which type of anesthesia is used, or composition of liposuction solution. But we concentrated on the pressure during liposuction. We used two types of negative pressure. Low pressure, only 200 torrs, or minus 700 torrs. It was low pressure, high pressure. Because it can be read in literature, some authors reported high pressure is worse for the quality of stem cells. But immediately after seeding, or in early cell culture intervals in primary culture, it is apparent stem cells isolated under high pressure adhered and grew better. You can see the cell population density. It stopped to work. The cell population density was by 1/3 higher, at least by 1/3 higher, than in cells isolated at low pressure. And this feature persisted even in first passage. We seeded equal amount of cells, and again we obtained by approximately 1/3 higher cell population density. And even in passage 2, it was the same. Thank you very much. Battery probably is run out.
You have the second knob on it. I put the same.
Second?
This is two knobs, and one is also.
But it's first.
Yeah, here it is.
First, but it is dying. Probably battery is run out, and the second doesn't work. It's dead completely. But here, the first button is good. Thank you very much. And even in after repeat passaging, the higher density or better growth of cells isolated under high pressure persisted, and we also tried in other patients, and the difference was even more apparent. Low pressure, bad adhesion, slow growth, high pressure, very good initial adhesion, and quick growth. So the cells isolated under higher pressure are better for growth, adhesion, and growth. And we also tried osteogenic differentiation. We stained by immunofluorescence stem cells for collagen 1, which is early marker of osteogenic differentiation. Alkaline phosphatase, it is medium marker, and osteocalcin, it is late marker of osteogenic differentiation. And we measured the intensity of fluorescence, and we normalized this intensity for cell number. And the result was, you can see, cells isolated under low pressure and high pressure. And in general, especially in alkaline phosphatase, in osteocalcin, you can see cells isolated under low pressure are better in osteogenic differentiation. They were worse in growth, but better in osteogenic differentiation. Collagen is similar because it's very early marker. All cells, practically all cells, are able to produce collagen, and you can also compare normal medium and osteogenic medium. Normal osteogenic, in osteogenic medium, the differentiation is more pronounced. As control samples, we used cell lines, commercially available cell lines, but they are not very, they are good for proliferation, but not very good for osteogenic cell differentiation. here you can see cell number, because we should count cells, and stem cells in general are more slowly growing than cell lines, and you can see again low pressure is worse, slightly worse than high pressure, and it was after repeat passaging and freezing and thawing the cells, and the difference still persisted. And we seeded finally, we seeded the cells, the stem cells, on metallic materials designed for bone implantation. It was titanium, aluminium, vanadium alloy, currently used in clinical practice. And we used several surface modification, grid blasting, rehashing. It was, in fact, milling and combination of both treatment. You can see all modifications are friendly for the cells, but under high pressure the cells react more sensitively to surface treatment. Proliferation was very good, similar to cell lines, but under low pressure the cells proliferated very slowly and were not very suitable for testing of materials. So we can conclude, as I told, cells isolated under higher pressure, minus 700 torrs, are better in cell adhesion and growth, and cells isolated under lower pressure are better for osteogenic cell differentiation. Thank you very much for your attention. You can see our institute.
So do we have any questions? Right.
Maybe a comment. It's very fantastic what you do, the different applications you show and how you test it. I would be very curious if you can use, instead of all the antibodies that you use so far, just Raman spectroscopy to very quickly give you information and give you also a spectra so that you can easily access the data that you have now. Maybe we should start to try.
It's a very good idea, but I didn't found in the Czech Republic Raman spectroscope suitable for living cells. We have broad collaboration with physicists, but they have usually conventional Raman. They told me it's not possible to observe living systems. Everything should be dried, and even, I don't know.
It's exactly why we invented this machine, so that
Yeah,
we can do all this nice
this machine
experiments
is not available, to my best knowledge, it is not available in the Czech Republic, unfortunately.
It's the first all around the world, so that's why we don't have it. But let's start a collaboration.
Munich isn't so far. Next.
Very exciting work. So, you show this high pressure will induce the cells better grow and later on better functions. Why? Is the cell stress cause that?
But we don't know exactly, because it's under investigation. But we have new results from flow cytometric characterization, and both populations, low and isolated under low and high pressures, differ. The population isolated under low pressure was more heterogenic, probably because the tissue is not so well disintegrated. We didn't isolate all stem cells, we isolate only some fractions which were variable, heterogeneous, and from this point of view, high pressure is better because macroscopically after liposuction, the tissue is more liquid, more different, more disintegrated, and after collagenase digestion, all cells, or most of cells are isolated, but in low pressure is bad. But in clinical practice, clinicians told me we never measured the pressure, but we think we use rather higher pressure than low. And it needs further investigation. We are in the beginning. We freshly obtained a grant with hospital, with Department of Plastic Surgery for this research, and maybe in next meeting I will tell you more.
Any further questions? Yes.
Have you, sorry, have you ever tried to treat cells selected with low negative pressure, with high negative pressure, after collection?
No, no, cells were treated with low and high pressure in hospital during liposuction, and then we have no chance to treat cells again with pressure.
Just what about the bulk?
Yeah.
It's enough.
we can try to treat them additionally. Do you mean to isolate them, for example, at low and high pressure, and then to treat them again with low and high pressure, or conversely, cells isolated at low pressure, we will try with high pressure, but probably it depends on primary isolation of different populations.
Maybe you just induce the secretion of exosomes or something like that.
Yes, it's good idea. I will try it in future.
If estrogen is a catalyst for differentiation, could it play a role in your low pressure cells to increase differentiation?
Yes, it's a good question, but generally in cell biology, it's very common that less proliferating cells, less proliferating but not dying, are more prone to differentiation. For example, if you have two rough materials, roughness is not cytotoxic. It only hampers the cell proliferation. And it is generally known very rough material with micro roughness stop cell proliferation and induce cell differentiation. And probably when the cells are not very high proliferation capacity and they have good conditions, no reason to die, and the cells are not lazy, they should do something so they're differentiated, but I don't have exact scientific explanation. We should investigate it more.
Well, it may be that all your lab techs that were working with the high pressure ones were female.
Yes, but gender is also important. But we have collaboration with Department of Plastic Surgery, and the vast majority of patients are women because men usually don't want to improve the organism by plastic surgery. 99%